Good evening, ma'am.
Good evening, Jose. Let's wait for a few more minutes before we start. Welcome to the last session of the NPTEL Live session, which is for plants and bioprocessing course. So, I think let's, I don't think we have to wait more than 2 minutes for some others to join. So,
Ma'am, not audible.
are you able to hear me?
Yeah, okay.
Yeah,
Okay now.
yeah. So, I have some few questions with me. So, we will discuss along the discuss answers for these questions. Um, so the first question is plant survivability is not affected by vegetation speed. Uh, the answers are true or false. What do you think is the answer for this question? So far we have discussed about suspension cultures, the effect of different parameters on suspension cultures. So by far, so you can guess the answer for this question.
Ma'am, I'm not getting you clearly.
So how does agitation
Yeah,
speed?
it's okay now.
Yeah. So how does agitation speed affects the plant cell's viability?
Hello.
Yeah, can you get your, am I audible?
Yes, ma'am.
Yeah, so like I was asking,
Uh,
yeah, how does agitation speed affect your plant cell viability?
Plant cell viability, I think it will be affected positively.
Positively, it will affect or not affect?
High agitation speed, I think it will shear the cells and all, then it will affect.
Okay, so then answer, can you answer the question? The statement is the
False.
plant cell, yeah, right, yeah, right. So, so Fauji, you're from this science background, right? You are not an engineer.
I am in my first stage of PhD, plant biotechnology.
Okay, so any idea about, so far we have discussed about cell suspension cultures and all. So have you ever seen cell suspension cultures?
I have seen it in some workshops and all, and I didn't work it alone.
Okay, so were you able to visualize things when the lectures were taught, like plant cell suspension, agitation, temperature and all, how it's affecting?
It is theoretically it is clear, ma'am.
Okay, okay. So, so if that is the case, I think you can answer these questions. So in plants and suspension cultures, the exposure time of elicitors can affect the secondary metabolite productivity. So I think you are on like this week
Yeah,
is all about case
yeah,
studies, right? So with the
yeah.
case study you
Elicitors
have
will,
seen, you can answer this
elicitors
question.
will induce. Yeah, it will induce the secondary metabolites
Yes.
production.
So the answer would be true, right, for this question.
Yeah, yes, ma'am.
So how do you calculate the exposure time, or how do you come to in terms with how much time you have to expose the elicitors? How do you do that? Any guess?
Um, elicitors.
Or at what during, say for example, you have a culture, you will be adding that in particular. This is the growth phase of your culture. Let's see. This is a lag, and this is a log phase, and this is stationary and death phase. So, in which phase of treatment you will be adding? Because in your case study, it is they were adding it in after the 8th day, right? So there would be a reason why it was chosen. Why not in the start itself? Why at the last or why at the middle? Something like that. So, what do you think?
Yeah, ma'am. In the stationary phase, I think the elicitors we are adding.
Yes, almost
Yeah.
before or when they are in their early stationary or end of the log phase, we will be adding your elicitors. And how long will you continue using it? So, say for example, your cell has reached stationary phase around 12th day and death phase is around 24 days. So, death phase is starting at around 20 days. So, your elicitor treatment up to how long? Like at what stage you will harvest your cells? If you keep, sorry. So, exposure time is all about how long the elicitor is being present in the cell suspension system, right?
Your voice is breaking, ma'am.
Sorry. Am I audible clearly now?
Yeah.
Yeah. So, um, how long do you plan to keep your elicitor in place? Like how long? See, for example, as you told, if you have added the elicitor at the initial stage of your stationary phase, so how long? So you will be harvesting your cells at which phase? So if you are adding your elicitor here, the concentration will be present towards your, like it will be there until you harvest your cells, right? So say for example, if your late log phase or you are adding the elicitor during this time, you will continue. Death phase will start at 20. So what you will do? Just before your deceleration phase starts, you will harvest your cells. So the exposure time would be around the day you harvest from the day you like initially added it. So say for example, if you harvested it around the 18th day means 12 to 18 days, the 6 days. So 6 days the elicitor is in the system, which will affect the secondary metabolic productivity. So that I was like trying to get the exact explanation of what is the exposure time of elicitors. So did you want? Fauzi, are you talking? I'm not able to hear you.
No, ma'am. No.
Yeah. So are you able to get it now?
Yeah.
So your answer was correct. I was just getting to know what are these, what does exposure time of elicitor mean in this statement. So what is productivity actually? Any clue? because the entire case studies was mostly in terms of productivity, right? Productivity, concentration amount, azadirachtin, productivity of azadirachtin. So, in a clue, what productivity is? Concentration is fine, it is your amount per volume. What is productivity?
How much amount of this one is produced?
Produced with respect to, it is with respect to time. So, say for
Yeah.
example, 100 grams you got in the sense for reaching 100 grams, how long it took? It took you only 20 days means 100 grams per 20 days. That will give you the productivity value. Okay, so give me 2 minutes. Okay. In the below expression, what does SM stands for? Uh, the equation is mu is equal to mu m 1 minus s by sm the whole of alpha. So please read the question.
Ma'am, are you speaking? We are not getting anything.
No, I have given time for you guys to read the
Okay, okay.
question. Sorry. So what is the answer for this question? Do you guys think? What does SM means? There are two components, right? There are two components, which is S and SM. If it is S means, what does it mean? Generally substrate. So what do you think SM stands for?
Maximum.
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 So what it will, because there are two options which has maximum in it. Option A and B. So in your case study, there was a part in modeling. So can you guys think any of it? Here actually it stands for concentration of substrate for maximum or complete growth inhibition. So did you guys go through the lecture? I think it is 28, where 28 or 29, I don't remember exactly. So where the professor was talking about modeling to predict the biomass and the azaractin content. So they were modeling, when they were doing modeling, kinetic modeling, they were including substrate inhibition at certain levels. So this equation comes in that part of your lecture. So why do we have to do modeling? What do you guys understood about modeling? Am I audible? Any idea? No. Sure. Let's go to the next question then. Okay, here comes again. So what were the advantages of mathematical modeling in the case study? So the options are offline simulation of nutrient feeding strategies and fed batch or continuous cultivation, enabled prediction of biomass growth or product formation and substrate utilization kinetics in batch cultivation. It could identify the optimum conditions of temperature, pH, and vegetation speed, etc. Could reduce the hit and trial experiments for improving azar-reactive productivity by azar-ending of plants or cultivation reactors. So you guys, if you have gone through the lecture videos carefully, so you will be able to answer this question. So the same question which I asked for the previous one. So what do you think is mathematical modeling and why? What are the advantages specified to this particular case study? So what do you guys think the advantages of mathematical modeling in this particular case study? Nobody remembers? Or you need time to recollect things? So I will give one answer. After that, I will expect everybody to answer. So in your particular case study, it was used for what and all it was used. It was used for offline simulation of nutrient feeding in fed batch and continuous cultivations. They used in model how it behaves and they got the predicted values and they did validation. So they did validation in experimental validation and then they were comparing how much the model predicted and how much experimental validation showed, right? So what were the other advantages of mathematical modeling with respect to this specific case study? Here are the options. There are two more answers here. They were able to reduce the hit and trial experiments for improving the productivity by plants of cultivation reactors. Right. What do you mean by hit and trial experiments? This is a general term, right? You guys can answer. What is hit and trial experiment? Okay. So in case you are optimizing something, say for example, um, say let's say temperature, pH, and vegetation speed. You can keep on doing automation. So for example, if you choose the RPM to be 50, you can select 60. If you do this, what is the productivity and the biomass? If you do this, if you set the RPM at 60, and what is the productivity and the biomass? Like that, you can keep on doing that. If you keep on doing that at one individual without any optimization, how can you arrive at, like, how long the experiments will take? How many times you have to how many variations of changes you have to make. So in order to avoid that, this modeling can be adapted, which will help in reducing the hit and trial experiments for any uh, plant cell cultivation or any other bioprocessing system. So in the particular to this case study, this answer is true. So also it enabled the prediction of biomass growth, product formation, and substrate utilization kinetics in batch cultivation. So they were able to visualize if they put this much amount of glucose and this much amount of nitrate and this much amount of phosphate, everything, and they will be expecting this particular amount of biomass growth, and the product will be up to this level, the substrate utilization will be at this rate. So when the predictions are made, it will be easy for the researcher or whoever doing the experiment to come in terms to form the or devise their experiment. So I think this is okay. So at the end we will have a small activity to discuss how to make it, how like we can set the experiments. So let's go to the next question. In the particular case study given, what could be the reason for improved volumetric productivity of azadirachtin and the batch bioreactor with centrifugal impeller than with the static impeller? So they have used two different impellers, right? One is static impeller, which is a modified, what you call, um, kind of restaurant type, a dish type impeller, and another one is a centrifugal impeller. So they got more productivity in what they got in the case study. The productivity was more in centrifugal impeller. So what do you think will be the reason for the improved volumetric productivity of azadirachtin? What happened? What was the difference between these two? So, so in the case study, what could be the reason for improved volumetric productivity of azadirachtin in the batch bioreactor with centrifugal impeller than with static impeller? Can you guys give me 5 minutes or 2 3 minutes? I will join. Also, uh, sorry, sorry guys, I need 2 to 3 minutes. What would be the answer for this question? So in that, you remember there was a table column where it was a comparison between your centric and centrifugal impeller. So there you can see the centrifugal impeller had better um, KLA. Like if there is the mixing time was less there, which is around 5.2 if I remember. Mixing time was less, and what else was there? It had better volumetric productivity. Like volumetric mass transfer coefficient was better. Like so the KLA then what mass transfer was better, and they were able to reach the desired concentration, and this centrifugal impeller had a lower mixing time. So the answer for this question is better KLA and lower mixing time. So the following question, please take few minutes. The steps to be followed to formulate a mathematical model are jumbled, evaluation and optimization. So here are the jumbled steps. There are 5 options. Please arrange them in the right order. The first one is formulation of model equation to describe the system behavior. Second one, you guys have to let me know. Second, third, and fourth. The fifth one is the simulation of experimental data. So, what is the answer for this question? What is two stands for? What is two means? what will come in that region? Please fill that. The options are above.
The second one will be determination of parameters.
Model parameters. Yes, right.
Yeah.
Third one would be
Parameter sensitivity test
mm, first you have
or
yeah.
yeah. mm,
So now only you have in the second step only you have set your parameters, right? So the next would be what?
Evaluation.
Evaluation. Yeah, it is evaluation. So
Mm,
now you are setting your parameter, then you are doing
and
evaluation and doing optimization of your
yes,
model parameters. So after
yeah,
which what you will do?
the determination of model parameters.
Mm, So why do you have to do that? Sorry, this is supposed to be this. Sorry, forget this thing. So this is first is determination. The third is what evaluation. So the at the end you are simulating. So before that, what you will do? You have to do parameter sensitivity test. Na? You have to remove the non-significant or insignificant parameters. You have to remove, and you have to choose on the only the significant parameters needed for your production. Is it right? You were telling that before, na? So the answer for this question is the fourth one is parameter sensitivity test. So what they do in parameter sensitivity test? So what is parameter sensitivity test? No idea. So, so let's do an activity, I think. So I will give you 10 minutes. In that 10 minutes, please develop or how can I tell? So I want you to uh, make a process. Say for example, choose a plant, or I will only give. So let's say, so I need uh, apple cell culture, apple cell culture, and what is the major or apple? What is majorly found in apple? So, okay, let's not talk about products. So I need biomass of this apple. So please develop apple cell culture. For that, what are the steps you will be needing? I want in a say for example 20-liter bioreactor. From the initial, which is your surface sterilization, to 20-liter bioreactor cultivation. Can you guys tell me in stepwise manner so that your entire understanding on your plant cell bioprocessing course will be good. So can you guys make a list of steps involved to produce apple cell culture at the level of 20-liter bioreactor? Can you guys do that? Please take 5 to 10 minutes. With that, we can end this session. Please take 5 minutes.
Ma'am, we are supposed to start from the
Yes,
surface sterilization
yes,
itself.
yes. How you will do? You will do
Mm,
surface elevation, then come to callus, then what you will do to reach
okay.
20-liter bioreactor? So done with that. So
Yes, ma'am. First, we have to select the explant.
yes, you are right. Yeah. So after which, what you will do?
Then surface sterilization.
Okay.
Then the media preparation.
Yeah. So surface sterilization media, everything comes. So once you do surface sterilization and keep your explant, wounded explant in your media plates or flask wherever, so you will get callus. Supposed to get callus if the necessary plant hormones are added in the right concentration necessary for apple cells to be induced. So after once you got your callus apple cell culture, so what you will do after which? So in your case study, what they have done, they have selected
And
one particular cell line. How did they select? How do they select? In your case study, it was there, na? They have collected Azadirachta indica seeds from different parts of India and then they develop calic cultures. From there, they have selected what? A cell line. Specifically, it was fast growing cell line. So, say for now, you have selected that fast growing cell line. Then, what you will do after that?
the cell suspension cultures we have.
Okay, after cell suspension is in flask you will initiate. After that, what you will do?
Then the organogenesis.
I just need
No.
apple cell culture, na? I don't
Yeah.
need
Okay.
organs.
Yeah. Yeah. Okay. Mm.
So, what you will do? So, you will scale it up, right? You will first do it in cell suspension culture flask. Maybe first in small flask. Later on, you will go with bigger flask. So, you will do batch kinetics. What is batch kinetics? Why do you even have to do batch kinetics? If you put that only, you will know how your growth. Say, for example, you are putting 2 grams in 50 ml media and taking one, say, for example, 6 flasks you have, and you are keeping it for, say, for 25 days. You have to take reading of one flask at a regular interval of time. Say, for example, one flask you will harvest one day, which is, say, for example, fifth day. You will take it out, take the flask out, remove the media out, remove the cells separately, calculate fresh weight and dry weight. Same way, 10th day, take another flask, 15th day, take another flask, 20th day, another flask, 25th day, like that. This is an example. So then what you will know, if this will tell you how the growth is happening. Say for example, if at my fifth day there was not much change in my biomass, so instead of from 2 grams it has grown only 2.1 or something, 2.34. So then what happens? Rapid growth stage, then your stationary and your death phase. So this curve you will get to know only when you do batch kinetics, which is nothing but depending upon the time how your biomass or cells are growing or varying in size. Okay, so this will give you a picture on when you have to inoculate and when you have to harvest. So once you know that, you can go to lab scale reactor. First, at what how many liters of reactor you will go. In your case study, how many liters they have used? Any idea? So first you will go with, say for example, 2 liter or 3 liter reactor. Next maybe 5 liter reactor. Depending Dependingly, you will be scaling up. What a scale up? What a scale up means? What a scale up means?
Wow. The amount of cell lines we are
Amount of?
no, the cell lines are we are collecting for.
No, no, no. What is scale up? Scale up means you are scaling the volume of, like, your different stages you are. Say for example, volumetrically you are scaling things up. Say for example, 3 liter reactor. First, if we are starting, first you started with small 250 ml flask. Say for example, then you went to 500 ml flask, then you went to 1 liter flask, then you went to 5 liter flask. This the order of increasing the volume that we call scaling the process up. Okay, so automatically what will happen here? You might having like the inoculum density would have around 5 grams. Then for 500 ml media, what you will do, or 500 in a 500 ml flask, say for example you had 100 ml media example, so almost around 10 grams you will inoculate. So if that is the case, the cells you are going to harvest is increasing. So what will happen when you scale things up? Will increase your biomass, will increase your product, everything increases. Okay, so this is the just how the steps. So if in case now you guys have generated, say for example, let's consider you have made suspension culture of which plant we can take, coffee beans, coffee bean. Say for example, Fausia has now developed suspension culture of coffee beans. She has suspension cultures of coffee cells. So, say for example, in the suspension culture, without any optimization, not optimizing anything, she is able to get caffeic acid levels of caffeic acid of 10 mg per liter concentration. She is able to get. Let's assume. So, she wants it to reach at least around 200 mg per liter. So, what she has to do to increase your caffeic acid in your coffee cells from suspension, you need 200 mg per liter. What you will do? What are the different ways you will follow to make it 200 mg per liter in suspension only? let's not talk about reactor. First, if in suspension itself, if you give me the like different ways you can enhance the production, same can be used there also. So first, let's discuss with suspension only. So, what are the strategies you will be employing to increase the production or product concentration?
Ma'am, more biomass. The
What you can do? So, you have more biomass also, it doesn't matter, like per liter calculation.
okay.
Yeah, so
Oh,
you
okay.
guys have, in your case study, have gone through, ma'am has talked about different strategies they use to improve the productivity or
Yeah.
concentration. So, do you
Okay.
remember? So, you can use elicitors if that is. So, when they used elicitors in Azadirachta indica, what happened? What you will do? You will do elicitor. Like if you add elicitors, what happens? There are high chances your concentration of the particular product is going to increase. Or still use, sorry,
Yeah, the mechanical stress that is
yes, that you can do.
agitation.
You can still optimize, sorry, 1 minute. You can optimize your conditions. Here I have not mentioned whether we have used optimized conditions, right? So
Okay.
this is before optimization, let's consider. So if you do optimization, what will happen? Optimization of your environmental parameters like pH, temperature, and what else is there? RPM, those things, and also your media components. Media components in the sense you can optimize your sucrose, optimize your nitrogen source, optimize plant growth regulators. There are multiple optimizations. So, say for example, if I do everything and found optimum values and I was able to reach up to, say for example, 40 to 60 mg per liter, after that, what I will do? With this optimized culture conditions, I have now one flask. So, for example, it is able to produce around 60 mg per liter of caffeic acid at specific RPM, at a specific pH, temperature, and at a specific concentration of different various components, media components. So, then what you will do, like you said, we can add elicitors, or we can add, what else have you studied? Precursors. What are precursors? What are precursors? So, precursors, what are they? Say, for example, caffeic acid is there means somewhere in their uh, previous steps in the pathway. So, these are intermediates of a metabolic pathway from where your caffeic acid is originating. So, this is like if you add something in the medium itself, the flux will be directed towards the production of that particular product. So, elicitors you can do, precursors you can do, mostly plant cell products are intracellular, so you can add permeabilizing agents. What they will do, if there is a cells, and they will cause small pores inside like on the cell surface so that your intracellular product can get leached out. Like it is easily available for further downstream processing steps. So this almost this like I think I have covered similar trends which were discussed in the case study. So we have come to the end of these sessions. You guys, if you have any doubts, please ask me. Uh, so when is the exam for this course?
It is 24th, ma'am.
24th. Okay, so it
Yeah,
is maybe in the next week, upcoming week.
yeah,
Yeah.
yeah.
So I hope you have gone through all the lectures carefully and taken enough notes. Hope you
Mm.
understood topics. Do you have any doubts?
Not now, ma'am. No, it's okay.
Okay, so if there are no doubts, we are at the end of the session. Thank you for joining.
Okay, thank you, ma'am.